Okay, dear class, let's collect what we've just found out. We have talked about the synapse today, and the question was what happens when an action potential reaches the synaptic terminal, and how is an electrical impulse transferred from one neuron to the next. So let's start. We have the electrical impulse, the action potential at the beginning. What happens to it? Okay, Dana, please start.
So basically, when an action potential comes near the synapse, Ca2 plus channels open and calcium diffuses.
Okay, thank you. Let's take a short look at our model. Here we have the synapse or synaptic knob. Here's the axon. The action potential travels down this axon and causes those Ca2 ions diffusing into this membrane. And what do they cause? Louis?
After this diffusion, the calcium binds the synaptic vesicles and they simply fuse with the synaptic membrane.
Yeah, with the presynaptic membrane. We can also see it in our model. Those little yellow bullets represent the vesicles, and they contain a certain substance which is important for the impulse to be transferred. And here they bind to the presynaptic membrane and they release something. What do they release? Yes, please, Daniel.
Yes, they release neurotransmitters, like for example acetylcholine, and these are released into the synaptic cleft. And after they reach the synaptic cleft, they bind to the receptors of the postsynaptic membrane, to some proteins, which are also known as chemically gated ion channels.
Okay, yeah, that's right. We have those chemically gated ion channels at the next neuron, at the so-called dendrites, which take up the impulse. And how does it happen? How is a new action potential created then? We have those channels. They are chemically activated by the neurotransmitters, by the acetylcholine, for example. But what happens? Fiona?
So as the sodium channels open, we have an influx of sodium into the postsynaptic neuron, and this causes a depolarization of the membrane, and thus the action potential is generated again.
Yeah, that's right. Thank you. Okay, let's sum it up in two short sentences. At the beginning, we have an electrical impulse which is converted into a chemical impulse by those neurotransmitters which are released into the synaptic cleft. Those neurotransmitters cause so-called chemically activated ion channels, sodium channels, to open. So sodium can diffuse into the dendrites of the next neuron, and this creates a new electrical impulse, so a new action potential which is created and sent along the next axon, which we can see here. So this is the direction of the impulse moving down the axon. Yeah, thank you very much. Let me just ask some questions. Here we have the model again, and you see here the orange one, the presynaptic membrane, those vesicles. Here is the axon. My question is, what are those little red structures? Sonia, please.
So these are mitochondria, and they are basically there to provide energy for the whole transport.
Yeah, that's right. That's right. You will find those mitochondria along the whole neuron in the axon at the synaptic knob, because everywhere where ions diffuse into the intercellular space, they have to be carried back in a way, and this is done by the use of so-called sodium-potassium pumps, for example, and this requires energy. Thank you very much. So my last question is, we have those acetylcholine, the neurotransmitter, binding to the receptors. If the new impulse is created and a new action potential is generated, which travels down the next axon, what happens to those neurotransmitters? Do they simply stay? So then the whole thing, the whole ion channel, would also stay opened. Dana?
So basically, they're broken down by acetylcholine esterases, and the products diffuse through the presynaptic membrane to be used again.
Exactly, right. And that's important to remember. They are reused again, because if they would stay at the receptor, there would be a constant excitation of the dendrites. That's it. That's it.